Hello, good morning, everybody. Welcome to today's webinar. First of all, let me thank you, all of you, for attending this webinar, but also for the speakers today. That for the next 2.5 hours, more or less, they will be talking about Mediterranean diet. From different perspectives. We'll talk about product development, we talk about digital solutions, but also you will hear today results and a little bit of insights regarding the MED4Youth project, but also you will hear about another project that shares common objectives with MED4Youth, which is Switch to Healthy. So without any more delay, I will present our first speaker, Professor Alfredo Martinez. Let me just give a couple of words regarding Professor Alberto Martinez. He is the director of the Precision Nutrition and Cardiometabolic Health at IMDEA Food in Madrid. He is also the co-IP, and he has been involved in several landmark intervention trials, such as Diogenes, Seafood Plus, Neo Food for Me, and PRIMA, whose results have been published in the most relevant medical and nutritional journals. He is also a supervisor of more than 75 PhD students, and he has published in more than 800 peer-review papers. He has been president of the FESNAT and the International Society of Nutrigenetics and Nutrigenomics, and he is currently the president-elect of the International Union of Nutritional Sciences. So thank you very much, Professor Martinez, for being here, and I give you the words. You can now share the presentations if you want, please.
Okay, thank you very much for your kind introduction. I am very pleased to participate in this EuroCat activity and initiative, and especially I wish to thank you, Professor Pedro Mena in the University of Parma, for inviting India to participate in this specific activity. Okay, I declare no conflict of interest concerning this presentation, and initially a few words about epigenetics. As probably all of you know, epigenetics involve heritable changes in gene expression that cannot be explained through variation in DNA sequences. Therefore, there are different phenomena related to epigenetic affecting gene expression. DNA methylation, or the additional removal of methyl groups, predominantly where cytosine is followed by a guanine, they call CpG sites. A second phenomena related with epigenetic is the post-translational non-covalent modification of N-terminal tails of histones that involves phosphorylation, somolylation, ubiquitination, acetylation, and methylation. In general, tightly folded chromatin, heterochromatin, tends to be shut down or not expressed or lowly expressed, while more open chromatin or chromatin is functional or expressed. The third mechanism of interest in epigenetic phenomena is related with microRNA. Indeed, microRNAs are single-stranded RNA molecules of 21, 23 nucleotides which regulate gene expression. As you can see there, a typical biochemical definition is that microRNA is complementary to a part of one or more messengers which may promote cleavage of the RNA and affect gene expression. In this particular view, to have a complete scope, epigenetic modification that can affect gene expression are those related with chromatin packaging alteration, DNA packaging around nucleosomes, or histone modification, DNA methylation, or microRNA expression. In this introductory presentation, I want to emphasize that the value of the epigenetics in the context of Mediterranean diet involve how dietary factors affect epigenetic modification and some disease. Therefore, some very simple examples: diet modified DNA methylation. You see there that a diet low in methionine, folate, and choline induce steatosis, liver fat accumulation, but also decreased DNA methylation in 2 different strains of mice. Another quick example is that a methyl deficient diet is lipogenic and also have a role in liver steatosis, altering the histone methylation pattern. Another example is that high carbohydrate diet may modify histone acetylation. An example related to Mediterranean diet is that resveratrol increase NAD-dependent histone deacetylase, HDAX. You see, in vitro model, there some impact on a specific deacetylase. Therefore, after this second stage, we have seen the definition of epigenetics. Then we have seen that diet can influence different epigenetic phenomena. Therefore, I am going to go further and incorporate the idea of Mediterranean diet related to nutri-epigenomics. You can see here diet and nutrients, the metabolic status, disease have impact on microRNA levels, in protein levels, in activity of specific enzymes that can favor or repress the expression. You see there a so-called histone code where repressed gene can be activated depending on particular, as you can see there, histone modifications. Just to refresh that epigenetics can occur in the adult, but also have a transgenerational transmission. In other words, maternal care, aging, dietary compounds, toxin, inflammation, regulate DNA methylation. This is in the fetus, in the early stage, in the embryogenesis, but also changes in DNA can occur during the adult life and mediated by, again, aging, dietary compounds, toxin, inflammation, and other exogenous factors. Today, in the time that has been allocated to this presentation, I would like to show you 4 examples where the Mediterranean diet is associated with methylation in specific genes. This is the case. We investigated the adherence to the Mediterranean diet with changes in methylation status on peripheral blood cells. This is the PRIMA study. Some of you are aware of this. There are 3 diets, and all of them receive different advice, different prescription to follow a given diet, particularly group 2 with olive oil and group 3 with nuts show that they increase the Mediterranean diet adherence a little bit higher than the low-fat diet that was used as control. The interesting point is that following a Mediterranean diet induced changes in inflammatory molecules like CPR, different CIM, in TNF-alpha and BCAN. This is a fact that the individual over the median, higher of the percentile 50 of the Mediterranean diet, after 5 years, they reduce the level of inflammatory markers. Also, it has been found in this study, in this research, that inflammatory canonical pathways are associated with different methylated CpGs. In particular, we see that the immune system genes related with T cell responses, with inflammatory response, with macrophage differentiation, with TFG signaling, or granulocyte response, show differentially mediated genes correlating with changes in the Mediterranean diet with 14 points. Going further in this investigation, you can see that there are positive and negative association of the methylation of inflammation of methylation of related genes with the adherence of the Mediterranean diet. In other words, some genes, you can just see as an example, leptin, as you increase the adherence of the Mediterranean diet, there is an increase in methylation. In some cases, the methylation is positive or could be negative or inverse when the adherence to the Mediterranean diet is decreased. And the value of this investigation is that the methylation of leptin, TNF-alpha, and CRP, three well-known inflammatory related markers, are associated with the methylation levels of some specific genes. Therefore, from this specific presentation, we can just conclude that the adherence to Mediterranean diet is associated with changes in the epigenome through methylation mechanisms in peripheral white blood cells in cell notable way in at least 50 genes, and some of these genes are related to inflammation. The second block of information related of the impact of consuming extra virgin oil versus nuts on DNA methylation, again in peripheral white blood cells. As you can see here, we analyze the intervention with this diet, and we analyze the methylation status in some specific white blood cell markers. You can see here the subject in the three diets, low fat control, Mediterranean diet with olive oil, Mediterranean diet with nuts, showed that the intervention has an impact on the consumption of these compounds. But more interestingly, we see that the consumption of olive oil or nuts have induced change in methylation and also in the delta methylation. But again, in some cases, we can see that the consumption of nuts or olive oil have impact on the differential methylation of some specific genes. And finally, and more clearly, the intake of polyunsaturated fatty acid was related with the methylation in this particular CpG. Therefore, for this second block, we can conclude that an intervention with Mediterranean diet supplemented with olive oil or nut is influencing the methylation of genes in peripheral white blood cells, and also that methylation changes occurs in genes related to intermediate metabolism, but also inflammation. The third comment is the quality of fat, polysaturated fatty acid, may be playing a role in epigenetic mechanism. A third example, quick example of, we measure the role of the low dietary folate intake. As you know, folate intake is increased in Mediterranean diet in the methylation of this specific gene. Very quickly, the consumption of folate have an impact on DNA methylation, and of course can have a subsidiary effect on metabolic alteration. You can see here the characteristic of these two groups. They have changes in the intake of folate, but not only in that, but in other functions. But again, if we look clockwise in this presentation, low folate and high folate have different levels of methylation. These methylation levels are related with insulin resistance, negatively affected. Then this OMA index as marker of insulin resistance is related with expression, and in the bottom left we see what we can just been hypothesized at the beginning that the methylation is down-regulating the expression of this gene. Again, a brief summary: subject with insufficient folate intake presented higher body adiposity and alteration in the metabolic profile, but this situation was associated with insulin resistance, and also the insulin resistance was affected by the expression of CMMKK2 gene. The last very quick example relates to the methylation with macronutrient intake. This is the group of CPGs that were selected, and very quickly, you know that the methyl data has very appreciated organoleptic properties and qualities. You see that fat intake, protein intake, carbohydrate intake is affecting some methylation patterns in genes related with olfactory receptors. To be the summary, again, nutriepigenetics literally means, in addition to change in genetic sequence, induced or mediated by nutrition. DNA methylation and histone modification are the two more investigated epigenetic phenomena related with nutrition. Take-home message: epigenetic marks can be modulated by the diet, and adequate intake of nutrients may be necessary for a healthy DNA methylation status, and I have shown that there are specific methylation roles of Mediterranean diet with quality fat and folate. This is one of the last. Epigenetics have a role. This should be discussed. What is clear is that epigenetics is related with dietary intake. In some cases, there will be causal effect. In some cases, will be a consequence of the diet. Therefore, we need to be care when we interpret methylation and epigenetic changes mediated by diet in relation with obesity, cardiovascular disease, fatty liver, etc. This is the idea, final, that this information, epigenetic, is very important for personalized precision nutrition, since there is no an optimal human diet, but there are different diets that depend on the genetic background, the family history, likes and dislikes, previous disease, culture, allergies, and physical activity, and of course, a precision medicine, a precision nutrition require also to consider, to interpret, to assess epigenetic marks. Thank you very much.
Thank you very much, Professor Martinez, for the presentation. Indeed, some of your relevant papers when we were designing the MED4Youth project that we will hear you about in the next presentation, we were following the conclusions that were from these studies. I don't know if there is any questions for Professor Martinez. I
can.
Yes,
I can ask,
maybe Tony.
Yes, thank you, Professor Martinez, for this nice presentation. I have a question concerning the methylation of the leptin receptor. I think, if I'm not wrong, I think that in response to the Mediterranean diet, there was an increase in the methylation levels of leptin receptor. I would like to know if this was translated into downregulation of the gene expression of this gene, and how this was related with the leptin levels in blood and the resistance to leptin that can occur in obesity.
Thank you. Indeed, your comment is complementary to my presentation. We have performed this experiment related to the methylation of leptin, and in animals fed with different fat contents in the diet. And at least in leptin, methylation of leptin is related with increased methylation with decreased gene expression. But this is not always the case, and also depends on the sites. Therefore, the rule is that in general, higher methylation is affecting a lower gene expression, but this is not always the case. Therefore, I suggest that everyone should analyze case by case, although I assume your comment that indeed higher methylation is habitually associated with lower expression. Thank you.
Thank you, Professor Martinez.
Okay, thank you very much again for Professor Martinez for being here, for hearing about epigenetics, and also if you can stay a little bit longer. Now we will move forward with the webinar, and we will indeed start now talking about the MED4Youth project. And to do this, I would like to introduce my colleague, Dr. Antoni Caimari. He is biologist and biochemistry and PhD from the University of Balearic Islands. He is indeed the director of the biotechnology area here at URICAT, which includes the nutrition and health unit and the omic sciences. He has provided scientific contributions on the health effects and the mechanistic role of bioactive food compounds, such as polyphenols or probiotics, in the field of metabolic programming, obesity, and related alterations. Dr. Caimari is the coordinator indeed and the PI from MED4Youth projects. He's also doing technology transfer activities and participated in more than 60 R&D projects of industrial research for private companies in the fields of nutrition, food, health, and omic sciences. So please, Dr. Caimari, you can go ahead with the MED4Youth introduction, please.
Thank you, Biotza. Are you seeing my screen?
Yes.
Okay. Good morning, everyone. I'm going to give you an overview about the MED4Youth project. Let me start by saying that obesity is a worldwide epidemic, and youth obesity is not an exception to this trend. According to the World Health Organization, 18% of children and adolescents aged between 5 and 19 years, more than 340 million, were overweight or obese in 2016. If we focus on the Mediterranean countries to which belong the partners of this project, Portugal, Spain, Italy, Israel, and Jordan, the prevalence of overweight and obesity at youth is particularly high, ranging from 20 to 36% concerning overweight, and being the prevalence of obesity higher than 10% in the 5 countries. As you know, youth obesity is a strong predictor of adult obesity, which has very a negative impact on health. Therefore, the increased adiposity that occurs in obesity can lead to the increase of oxidative stress and low-grade chronic inflammation, which in turn can trigger the appearance of different alterations such as hypertension, fatty liver, insulin resistance, and dyslipidemia. These disease risks are factors for the development of diabetes and cardiovascular disease, which cause millions of deaths each year. Therefore, there is an urgent need to combat youth obesity to minimize the negative short and long-term health consequences. In adults, the clinical intervention study Prevención con Dieta Mediterránea, the PREDIMED study, which was previously commented by Professor Martinez, which was carried out with more than 7,000 middle-aged and elderly subjects from Spain, clearly demonstrated that the Mediterranean diet, enriched with extra virgin oil, olive oil, or nuts, was associated with a reduction of the risk to suffer cardiovascular disease and deaths compared to a low-fat control diet, which is the standard diet used for the prevention of cardiovascular disease. In addition, the PREDICT MED Plus study reported that a nutritional intervention based on an energy-restricted Mediterranean diet was able to decrease adiposity in addition to improve different factors for cardiovascular disease in middle-aged and elderly subjects with overweight and obesity. Concerning adolescents, some cross-sectional studies showed that the high Mediterranean diet adherence was associated with a decreased risk of being overweight and obese in adolescents from different Mediterranean countries. However, these studies were not clinical nutrition intervention studies in which the adolescents were assigned to a Mediterranean dietary intervention. When we wrote the MED4Youth proposal in 2018, only one pilot nutritional intervention study carried out with adolescents and children were published. In this study, it was demonstrated that Mediterranean diet was able to improve BMI, glucose and lipid profile in children and adolescents with obesity and metabolic syndrome. However, this study was carried out only with 49 subjects. Therefore, the effects of Mediterranean diet on obesity and risk factors for cardiovascular disease through well-designed clinical intervention studies and the underlying mechanisms were scarcely evaluated in adolescents with obesity before the beginning of the MED4Youth project. Thus, robust clinical data for the Mediterranean countries were lacking at the moment that we started the MED4Youth project. The first objective of our project was to demonstrate that an intervention based on an energy-restricted Mediterranean diet was more effective against obesity and the risk factors for cardiovascular disease than a conventional energy-restricted low-fat diet. The second objective of MED4Youth was to elucidate where the Mediterranean diet can shape the gut microbiota and the gut-derived metabolites, and to gain knowledge about the mechanisms by which Mediterranean diet exerts its beneficial effects against youth obesity and associated cardiovascular disease risk factors. To address these two objectives, the consortium of the MED4Youth included 6 partners with complementary knowledge. The clinical nutrition interventional study, as well as the analysis of the data, were carried out by the universities of Coimbra and Parma, as well as by EURECAT. The consortium also includes an Israeli company called CDTI, which provided the pomegranate juice and the chickpeas included in the Mediterranean diet. And the Spanish company Novapan, through its Spanish shop brand, produced and supplied the sourdough breads that were consumed by the teenagers. The Scientific Food Center from Jordan was in charge of the composition analysis of the food items that were included in the Mediterranean diet. To achieve the first aim, we carried out a multi-center nutritional intervention study aimed at including 240 adolescents aged between 11 and 17 years with overweight from Spain and obesity from Spain, Portugal, and Italy. The main outcome of the study was the BMI Z-score, expecting a significant decrease in the Mediterranean diet group versus the low-fat group. The study participants were randomly assigned to an energy-restricted low-fat diet, highlighted in blue, or to an energy-restricted Mediterranean diet, highlighted here in red. The energy-restricted Mediterranean diet was enriched in sourdough bread, hummus, pomegranate, and mixed nuts, which are rich sources of different bioactive compounds such as polyphenols, fiber, and monounsaturated and polyunsaturated fatty acids. A calorie restriction of 20% was applied, as was previously recommended for dietary management of obesity. Different anthropometric, clinical, and food intake biomarkers were also analyzed. Dietary intake was evaluated by 3 days dietary course, and the adherence to the Mediterranean diet, as well as the physical levels, food habits, quality of life, and socio-demographic data were also collected using validated questionnaires. The nutritional intervention was accompanied by an educational web application designed to encourage healthy behaviors to the adolescents. Interestingly, in this project, there was a follow-up period of 4 months in which the dietitians did not give any advice to the adolescents. The follow-up visit allowed us to know if the adolescents were capable, without the help of professionals, of maintaining healthy behaviors. To quantify this, the different questionnaires about lifestyle habits as well as anthropometric parameters were analyzed 4 months after the end of the nutritional intervention. Highlighted here in green, in the case of the Mediterranean diet, and in red for the low-fat diet, you can see the main differences between diets. The use of virgin olive oil was higher in the Mediterranean diet group compared to the low-fat diet group, and the consumption of cereals, bread, pasta, and rice was higher in the low-fat diet group. In addition, in the Mediterranean group, these specific food items, nut, hummus, pomegranate, and sourdough breads were included. Mediterranean diet was characterized by a higher consumption of total fat compared to the low-fat diet, mainly monounsaturated fat, whereas the low-fat diet was characterized by a higher consumption of carbohydrates, mainly of complex origin. And although a 20% of calorie restriction from total energy expenditure was planned initially, this percentage was adapted depending on each adolescent. We made personalized meal plans for the teenagers by providing them with a list of healthy foods for each food group, and they could choose the foods they liked to design the weekly menus. There was also a motivational interview at B1 at the beginning of the intervention with the adolescents and their parents to promote healthy lifestyle habits, which was accompanied with the delivery of a booklet including information related to these issues. In addition, the adolescents of both groups and their parents had access during the intervention to an educational web application. This will be explained in detail by Perla Degli in the next presentation. The second objective of the project was focused on the elucidation of the mechanisms by which the Mediterranean diet exerts the effects on health. Firstly, both metabolomics and metagenomics approaches were carried out in the feces of the volunteers, and afterwards, most of the data obtained in the study were integrated using a system biology approach. These approaches will be explained by Elena Torrey in the next presentation. My last slide is focused on the flow chart of the intervention study. As you can see here, we included finally 148 volunteers who were randomized to receive the Mediterranean diet or the low-fat diet. I would like to highlight that the sample size was about 62% of the was initially planned, which was 240 subjects. No? During the intervention, there were 35 dropouts, which was 23.6%. This was in line with the expected dropout, estimated to be 20%. And at the end of the intervention period, at V3, we had 113 volunteers, far from the 192 that were initially expected. The two main reasons of this lower number of participants were the COVID-19 pandemics, as well as the target population, which is usually more difficult to engage than adults. This lower sample size probably had an impact on the expected outcomes, as you will see in next presentations. Thank you very much for your attention.
Thank you very much, Dr. Kaimari. As just remember, you can pose your questions in the chat. We will be trying to answer those possibly directly on the chat, or if not, then after the different sessions we will also can have a more open discussions building upon them. So thank you again, Dr. Kaimari, for the overview of the MED4Youth and for introducing different of the building blocks that now in the next presentation we will hear about. But before moving to these building blocks, let me also introduce now our next speaker, Simona Mincione. Okay, Simona, she's a EU project coordinator of the Switch to Healthy project. She's a senior project manager at ENCO, and she has great experience in EU and R&D grants and project management. She's a seasoned professional with a master's degree in political science and also holds a certificate from the World Intellectual Property Organization on IP management. Since more than 10 years, she has been performing at senior project manager and playing a pivotal role in overseeing different projects. With extensive experience in EU research and development grant, she excels in project management, contributing to the advancement of innovation projects. So please, Simona, please tell us about the Switch to Healthy project.
Yes, good morning, everyone. First of all, thanks a lot to Eureka team for inviting us to introduce our project. Thank you for all the guests to joining us today. This is a journey actually that we want to introduce you today. And you see here, our mission is very, very clear. The Switch Mediterranean, we want to switch Mediterranean consumer to Mediterranean sustainable dietary partners. And we want to make a significant impact complementary to MED4Youth action. So let us now collectively explore how we are contributing to well-being of family across Mediterranean diets. So today, our narrative is more or less structured in 3 key points. I will give you a little introduction about the Switch to Healthy project, and then we navigate into the concept that is at the basic of our action, and then to give you an introduction about our methodology, and of course, we are available for a Q&A session later. So before diving into details, now the essence of Switch to Healthy project, it is a project that is around 3 million, that it received around 3 million euro contribution from PRIMA, that is the program for research and innovation in the Mediterranean area. And it's, well, we can say that Switch to Health is not only a project, really a 36-month action, long innovation action that kicked off in April 2022. And we involve 80 countries from the Mediterranean area, uniting a consortium of 18 partners. And just today, we reached the final month 20 of our journey. You can see here the multidisciplinary consortium. We united experts from nutrition, from behavioral change, but also from innovators in food sector, engaging directly small and medium enterprises, larger enterprises. Today we have a representative from della, pioneers in IT and digital solution, and today we have CERT also with us, and association representing our community actually in the food sector. So we bring both private and public organizations across the Mediterranean area in order to collaborate in this R&D interdisciplinary initiative. Now, let's join into the heart of our Switch to Healthy concept that is that aspire to promote Mediterranean diet. Indeed, we want to increase sustainable adherence of our target group that are families, and so encompassing adults, but also kids and adolescents. And we want to promote and fortify healthy dietary habits. And you can see here how structured is our idea. So we start from data collection. We have a section totally dedicated on innovation and development. We have then family approach intervention. And then this is something that I'm really proud of, is about the creating an enabling environment. So all the results that we are generating in our project will be key for creating an environment where all the results can make really a change in our communities. Now, that you know about our concept, you can now learn about our methodology, so how we want our roadmap that guide us to achieve our impact. And you can see that this is structured into three main assets. So we have the citizen engagement and co-creation, the more creative phase, that is development and product and tools, and the phase regarding the deployment and the assessment. So first of all, regarding this collaborative effort that we do with families and citizens, we are organizing and getting engaged families from Spain, Turkey, and Morocco in order, first of all, to learn about their dietary habits, so through surveys and focus group, and to have a co-creation with them in order to develop healthy snacks and participate in all the activities related to the co-design of educational materials and tools intervention. The second step is about, as said, the more creative one, the development of food products, so the snacks. And today we have Della that is going to have a fully detail about our innovation there, and the tools, the digital tools, materials. And so we combine these different solutions really to create a behavioral change going to the daily life of families. So this is something that the families, adults, so parents, kids, and adolescents can really use. And the third phase is regarding the deployment. As I said, we have direct engagement of more than 3,600 families from Spain, from Turkey, and from Morocco, in order not only to see how the different habits, but also the different culture and the different tradition, and also the different target age are impacting on, first of all, on the behavior, on the lifestyles, on the habits, and now we can really create a change there. And of course, all of this will be rigorously assessing the health, economic, and environmental impact, including, like, for example, the food waste impact that we are also assessing there. So our commitment is not just to initiate a change, but to really also measure the tangible effects in a very holistic way. So you have my contact here. I think that I give you some inputs and insight over our action. So I'm available to respond to your question or curiosity in the QA session later. Thank you so much.
Okay, thank you very much, Simona, also for giving us a little bit insights on the switch to healthy that indeed shares a lot with MED4Youth, and you are also facing the engagement of families and try to make this long lasting and really impact. I think you are still not results from the intervention itself.
We just kick off this phase. We have just launched the intervention phase this month. So all the families are already engaged. So this was really the crucial month where we start with the first, so M0 assessment, so we can really in 1 year, assess the change that we made through our solution.
Okay, yeah. So yeah, indeed, it's a very, very nice now period of the project. So also just remind to the audience that in case later on, if you also have any questions that maybe didn't come up to your mind, or that you want additional information, you will give you also an email, and then we will redirect your questions to the different speakers. I think there's one hand raised. Maybe better if it could be typed. But if you couldn't type the questions in the those that are raising hands, if possible for you, please to type them briefly into the chat, and then we can take progressively. There is one regarding educational material. Okay, and then we will we'll get back to all of them at the end of the session. Okay. So thank you very much, Simona. And so now we move indeed to starting to up to now we've seen overviews from the different projects. Let's start to get into results, into the different developments of the different projects, and we'll start with the results from the MED4Youth project that indeed finishes after 4 years due to extension from the pandemic. It ended now in November 23. So our next speaker is Dr. Alice Rossi from University of Parma. Alice, she is assistant professor in human nutrition at the Department of Food and Drug at the University of Parma. She has a master's degree in human nutrition from the University of Milan, a PhD in food science from the University of Parma. Currently, she is carrying out several European and national projects focused on eating behaviors and food choices, diet evaluation in terms of nutritional adequacy and environmental impact, nutrition education, with a particular focus on working with children and young people. So please, Alice, let us know about the result from the MED4Youth project.
Yeah, thank you very much, Piazza. Okay, just a second. Okay. Can you see my presentation now?
Yes, perfect.
Okay, thank you. So thank you for your kind introduction, and I'm very happy to be here today presenting the main results on behavioral and dietary data after 4 years of hard work. So this is a main achievement. As you already seen in the previous talk presented by the project coordinator, within the MED4Youth project, we carried out a multi-center randomized controlled trial enrolling adolescents from Portugal, Spain, and Italy having obesity or great overweight. Once enrolled, participants were randomized into one of the two dietary groups, so the MED groups and the low-fat diet groups. And the two groups differed in their nutritional composition. In particular, the MED diet was designed based on the diet used in the PRIMA study, so it provided higher amount of fat and a lower amount of carbohydrates than the low-fat diet. But the caloric content was calculated in the same way for all the participants, reaching up to 20% of energy restriction, depending on the characteristics and the nutritional needs of each participant. Therefore, for some adolescents, caloric restriction lower than 20% was applied, and in other cases, the caloric restriction was not applied at all because we set a minimum value for energy in order to avoid macro and micronutrients deficiencies. In addition, as we see before, participants in the MED group receive also typical Mediterranean products. So regardless of the diet group, participants receive a weekly dietary plan based on their nutritional needs and preferences. And participants in the MED group also had detailed instruction on how they should include the study products and the related frequency of consumption. Here you can see an example on how the products of the study are included also in the weekly dietary plan each day, and of course more times per day every day during the week. In addition, we also include a food exchange system. So we provided a list of food exchanges that allows participants and their parents and their families to choose the foods they prefer, to create their weekly menus in a more flexible way, but at the same time respecting the dietary prescription of the project. So the dietary plan could be accessed also via web application specifically designed for this project, through which it was also possible to access educational content to increase the knowledge of participants and motivate them to follow the intervention, as my colleague Perla will present in a while. And during the 4 months of intervention, we collected several information from anthropometric to behavioral data, as well as blood, urine, and feces samples. But now I will focus only on some of these data, and in particular on behavioral data. And the other will be presented in the following presentation. So first of all, here you can see the baseline characteristics of the 113 participants who completed the interventions phase. They mainly came from Italy, and they were equally distributed between intervention groups and genders. Then, in relation to the dietary or the eating habits, we initially focused on the analysis of the adherence to the Mediterranean diet using the KidMed questionnaire. And here, of course, it was a very hard work and was a teamwork. So I would like to thank Perla, Federica, Joanna, and Elena that work with me not only in collecting data, but also in analyzing data and prepare this presentation. So thank you very much. And at the beginning of the study, participants show a medium adherence score in both groups that increased from baseline, but without significant differences between visit 2 and visit 3 and between groups. However, we observed a significant change in the distribution among times, with participants moving from a low, medium to high level of adherence to the Mediterranean diet, but only in the MED group. Then, considering some specific answers of the KidMed questionnaire, we observed an increase in the distribution of participants consuming specific healthy food groups, as in the case here of legumes that increase in both dietary groups, and of nuts, but only in the MED group, resulting also in a higher percentage of participants in the Mediterranean diet than in the low-fat diet group that consumed nuts regularly at visit 2 and visit 3. On the contrary, we observed a trend of decrease in the consumption of other products like the sweets and the desserts, significantly, that was significantly only in the low-fat diet group for a dessert at breakfast. Then we also collected information about the habitual consumption of the food groups through a food frequency questionnaire that was validated within the European study ELENA, Healthy Lifestyle in Europe by Nutrition in Adolescents. And I've given here you just some examples of specific food groups. And here we have the habitual consumption of fruit and vegetable, We observed a trend of changes in the distribution of both participants of both diet groups for both fruit and vegetable consumption that seems to increase over time, even if not significantly. Similarly to what observed through the KidMed score, we also participants seems also to decrease their frequency of consumption of sweets and candies in both dietary groups, but again here we did not reach the significance. Then, in addition to the behavioral data, we are also analyzing the actual differences in the diet of the subject in the two groups thanks to the analysis of the food intake biomarkers. And this part of the work was led by my colleague, Dr. Claudia Favari. So thanks, Claudia. And we have monitored more than 200 compounds, identified more than 100 compounds, and quantified so far 39 of them, mainly linked to plant-based food intakes. If we move discussing about biomarkers of plant-based food consumption, here you can see as an example that we found a main effect of the dietary intervention in the excretion of the isoferrulic acid-free glucuronide that also decreased over time in the low-fat diet group, and of the hydrocaffeic acid-free sulfate. Then, similarly, for other compounds as 4-hydroxyhypuric acid, ferulic acid for glucuronide, and ferulic acid for sulfate, we found higher value in the MED group at the end of the intervention compared to the baseline. So, if we analyze altogether these compounds considered biomarkers of plant-based food intake, as we can see here in this graph, the amount was higher at the end of the intervention in the MED group, underlying a higher consumption of those food groups in the MED groups than in the low-fat diet group. We also analyzed phenolic metabolites, and this part of the work was carried out in particular by Nicole Tosi. And the phenolic metabolites analyzed, we analyzed in particular those derived from the consumption of the specific food that given in the Mediterranean diet group. And we were able to identify more than 100 metabolites, of which 88 have been also quantified. So, the quantified metabolites could be grouped in as specific metabolites from Mediterranean intervention food and as other more generic compound. And as you can see here, the amount of the specific polyphenol metabolites increase in the MED group, indicating higher consumption of those products provided to participants enrolled in the MED group. Then, considering specific compounds, here we reported some examples. So we found 3 different phenyl gamma valorolactones, which are colonic metabolites from flavanthiols, so linked to the consumption of nuts, that increase in the MED group after the intervention. Similarly, the enterolactone glucuronide and more in general the class of enterolignans increase in the MED group at the end of the intervention, probably due also to the consumption of the sourdough bread. Also, the urolithin A glucuronide was higher in the MED group than in the low-fat group, while the urolithin B glucuronide increased in the MED group after the intervention. And these compounds are probably linked to the consumption of pomegranate and of walnuts elagitannins. Then, we also collect other kind of information. So moving on other the data beside the dietary ones, we monitored another important behavioral aspect that is the physical activity level of our participant using the PAC-A questionnaire. So as you can see here, participants reported a medium-low physical activity level at the beginning of the study, with no changes over time or between groups, considering the physical activity level score, but also when considering the distribution of participants among the physical activity level categories. Last, since during the intervention, we had also an educational intervention for participants enrolled in both dietary groups, we assessed the nutritional knowledge using the knowledge questionnaire validated within the ELENA European study. Also in this case, we did not find any differences between groups and among times for the total knowledge score and for the specific subscore of the questionnaire, except for a specific one related to the energy intake and energy metabolism that resulted different between groups, but only at visit 2 and not at the end of the intervention, so with no differences in the total knowledge of participant. So to conclude, based both on auto-reported consumption data and on more objective measures as the biomarkers of food intakes, these results suggest that our intervention improved the eating pattern of participants, in particular in the MED group, resulting more in line with the principles of the Mediterranean dietary pattern. While the interventions did not produce any change in the physical activity level and in the nutrition knowledge of our participants. So that's all. I would thank very much all my colleagues from the Human Nutrition Unit of the University of Parma, all the students involved in the data collection and analysis, and of course all the partners of the MED4Youth Consortium. And thank you for your attention, of course.
Thank you very much, Alice. For starting to give the first results from the MED4Youth project, and if it's okay, we will to complete also an overview of the different results and then to wrap it all together later on. We move to the next speaker, Paolo Matafome. Paolo, he holds a PhD in biomedical sciences since 2012 from the Faculty of Medicine at the University of Coimbra. He is a researcher at the Faculty of Medicine at the University of Coimbra and invited auxiliary professor in the Polytechnic Institute of Coimbra. He has published more than 82 research articles and 3 book chapters, and he has participated in more than around 20 research projects as principal investigations and also as co-principal and team member. So Paolo will continue giving insight on results from the MED4Youth. So please, Paolo.
Good morning to all, and thank you, Beata, for the introduction. I will share my screen. Can you please confirm that you are seeing my presentation?
Yes.
Okay. So just to move forward after the presentations made by my colleagues here in these projects, I will show you the main results regarding the typical or the classical anthropometric measures related to obesity, and also to some biomarkers of adipose tissue function, also general metabolic markers, and oxidative stress and glycation markers. Just to remember what was shown by Dr. Kaimari, at the first visit and the second visit and the third visit, the patients were analyzed for classical anthropometric measures like the systolic and diastolic blood pressure. Also, let me show you the laser, heart rate, the BMI, the waist and hip circumference, as well as the waist-hip ratio, and also bioimpedance analysis of fat, fat mass, muscle mass, and fat-free mass. We then calculated the delta for each of these parameters, which means the difference between the two visits, the V2 and V3, regarding or in relation to the V1, and we used repeated measures and cohort analysis to analyze the changes between the different visits. And what we have seen is that for the blood pressure, both systolic blood pressure and diastolic blood pressure, as well as for the heart rate, we didn't find any major differences between the visits for both the low-fat diet and Mediterranean diet interventions. For the BMI-Z score, we found a significant effect of time, which was similar for both interventions, showing that both of the diets led to a similar reduction of BMI, in this case the Z-score. This was not observed for the waist and hip circumferences, as well as for the waist-hip ratio, which also didn't change. We also made the bioimpedance analysis for the muscle mass, the fat-free mass, and also the body fat percentage. And we found for the free fat mass a significant effect of time, but this was not statistically significant for the differences between each of the visits. So it means that with the both interventions, the participation in both interventions leads to a significant reduction of fat-free mass, but this was not observed for a specific visit. We then move forward to the classical biomarkers of health, and at V1 and V3, we collected blood samples and also urine samples for the determination of glucose, insulin, the calculation of the insulin resistance index, OMA, and also to the determination of the lipid profile, adipose tissue function markers such as adiponectin, leptin, resistin, and the ratio between adiponectin and leptin, which is understood as a ratio of adipose tissue function, and also inflammatory markers and oxidative stress markers, ages in plasma and erythrocytes, and also trimethylamine oxidase, which is a marker of gut dysregulation. So just to make a really fast overview of the methodology, the glucose and lipid profile were analyzed in serum samples following hospital routine analysis. All the determinations made by ELISA, including insulin, all the adipokines, it means the factors released by the adipose tissue, inflammation markers, and also glycation markers were made in ADTA collected plasma. The erythrocytes fraction that was on the bottom was then resuspended in ultra-pure water and used to evaluate the glycation markers and also enzymatic activity in erythrocytes. Then in urine samples, we analyzed the oxidative stress markers. Looking to the data that we got from these markers, we have seen that the fasting glucose levels were increased in the V3 of patients consuming the low-fat diet. And this was related with the downregulation of triglycerides only in the same group, which may be related with the type of diet, which was a low-fat diet, and the elevation of glucose may also be related with this, although we don't have a final conclusion about this. No differences were observed for insulin and also for insulin resistance. The total cholesterol was reduced in a similar way in both interventions, as you can see here and here. And no differences were also observed for HDL and LDL cholesterol. We then move forward to analyze the adipose tissue function markers. Adipose tissue dysregulation is associated with the development of low-grade inflammation in the adipose tissue. This is related with the development of insulin resistance and also an impairment of the adipose tissue secretome, which means that the adipose tissue reduces the expression of beneficial and anti-inflammatory adipokines like adiponectin, and increases the production of pro-inflammatory adipokines like TNF-alpha, MCP1, EL6, and also resistin. This leads to a propagation of this pro-inflammatory signal that is related, as I said, with the progressive development of insulin resistance. This is related with the fact that adipose tissue expansion may be associated with chronic hypoxia and also impaired angiogenesis, and then this leads to unresolved inflammation and impaired adipose tissue adipokine secretion. Following this rationale, we measured the levels of adiponectin, leptin, and resistin, and also we calculated the adiponectin to leptin ratio. And what we have seen is that although we don't have differences for leptin and resistin, and for the adiponectin-leptin ratio, we have seen a positive effect of Mediterranean diet only in the group consuming the Mediterranean in this type of diet. At the V3, we found an upregulation of plasma adiponectin levels, and there was also a downregulation of resistin levels in both groups. Then we measured the levels of pro-inflammatory factors like TNF-alpha, EL6, EL8, and IL-8 and MCP1, and we also found a specific reduction of EL6 only in the group consuming the Mediterranean diet at V3, it means at the end of the study, and we didn't find any other changes, namely in CRP and TMAO. Then we also were interested in analyzing the oxidative stress and glycation markers. And to analyze the glycation, glycation means the non-enzymatic glycosylation is produced through the reaction of glucose and other intermediaries of the glucose metabolism with amino acids from the proteins. And this leads to the formation of advanced glycation end products. And one of the main intermediaries of the glucose metabolism is methylglyoxal, which leads to the formation of a specific group of AGEs, including MGH1, arch-pyrimidine, and other different AGEs that are present in lower concentrations. This methylglyoxal is usually detoxified by the glyoxylase system composed by glyoxylase 1 and glyoxylase 2, which is dependent on glutathione and detoxifies the methylglyoxal to D-lactate. So the levels of methylglyoxal and the activity of the glyoxylase system are important markers of the glycosylative stress, which means the reaction of glucose with amino acid residues and the modification of these amino acids. So we, and sorry, I forgot to tell this, basically this glycation may happen in the circulating proteins, also in erythrocytes, in the extracellular matrix, and this can lead ultimately to the generation of oxidative stress in cells. And that's why it's important to measure the activity of these detoxification mechanisms in erythrocytes, because they are the first signal of increased glycosidative stress in the blood. So we measured the levels of MGH1, which is a methylglyoxal specific derived advanced glycation end product, and CML, carboxymethyllysine, which is specifically derived from glucose reaction with amino acids, in this case, lysine. And we didn't find in the plasma any kind of changes between the groups. And we didn't find any effect of both diets at the end of the intervention. We also measured MGH1 levels in erythrocytes, and we also didn't find any differences. In erythrocytes, we also measured glyoxylase 1 activity, and we found a specific reduction of the activity of this enzyme in the group consuming the Mediterranean diet. This means that the glycosidative stress is usually the signal for the upregulation of a compensatory upregulation of glyoxylase 1 activity. So the downregulation of the activity probably suggests that these patients had lower glycosidative stress. This was also found for the oxidative stress markers, namely 8-OHDG, which means 8-hydroxy-2-deoxyguanosine, which is a product of oxidative damage of guanosine, which was tendentially reduced by both interventions, but we didn't get any statistical significance. And we also observed downregulation of F2 isoprostane, or 8-isoprostane, which is a chemical modification of arachidonic acid induced also by oxidative stress. And this was downregulated in both interventions at the end of the study. So just to conclude this data, we have found minor changes in the anthropometric measures, only a reduction in the BMI-Z score, and this was similar in both interventions. Also, a similar improvement of lipid profile in both interventions, which was characterized by a reduction of total cholesterol. We only observed a difference in triglycerides and glucose in the low-fat diet, but as I said, this may be related with the specific composition of that plan. We can also say that the Mediterranean diet further improved the adipose tissue function and inflammatory markers, because we observed an upregulation of adiponectin and a downregulation of IL-6. And both interventions also reduced lipid peroxidation, but Mediterranean diet seems to have further effect in reducing the glycosidative stress in erythrocytes. So just to finish, I would like to thank to all the partners in the project that were amazing, and specifically EUROCAD test coordinators were amazing in conducting the project. Also to all the team at the University of Coimbra that was involved in all the steps of the project. And also, I would also like to say that this project is also linked with another project that we are coordinating here at the University of Coimbra, which also involves EUROCAD, which is the PAGHA project, a $9.8 million project funded by the Horizon Europe, that also aims to use the Mediterranean diet to tackle obesity. And we are, of course, open to future collaborations in the scope of this project. So thank you very much for your attention. And now I'm ready for questions that may have.
Thank you very much, Paolo. If it's okay for everybody, we will take the final presentation, and then we have already a couple of questions that have arrived to to us. So before moving to session 2, we will do the a QA. No, so we try to solve already questions that are on the table. But yes, first to close this first session of the agenda, my colleague from Eureka, Terena Torrell, she works as researcher in the genomics at the Center of Omics Sciences at Eureka, and she's leading the genomics projects. She holds a degree in biotechnology from University Rovira i Virgili and a master's in biomedicine from the University of Barcelona and PhD in biomedicine from the University of Rovira i Virgili. Her research experience has spanned various research areas, including molecular biology, genetics applied to health, biology, and the study of microbiota in both the intestinal and the environmental context. So please, Elena, she was going to talk about omic and data integration.
Thank you, Beata, for your kind introduction. And good morning, everyone. Today, I'm going to talk about omic sciences and how they play a role in the MED4Youth project. So I will divide this talk into 3 parts. The first one is focused on what omic science are. The second one on the main omic findings of the project. And finally, on the results of data integration. So, okay, what are omic sciences? The omic sciences refer to a set of scientific disciplines that involve the comprehensive study of biological systems at various levels and following the central dogma of the molecular biology. The central dogma outlines this sequential transfer of genetic information from DNA to RNA to protein and finally to metabolite. So we have to think that our DNA is like a master instruction manual telling our bodies what to do. RNA take those instructions and turn them into proteins, which are the functional molecules. And these proteins interact with substrates and products, which we call metabolites. So we've got these principal branches of omic sciences. Genomics look at genes. Transcriptomics studies RNA. Proteomics studies proteins. Till we get to metabolomics that studies the metabolites. And there are so many more. We also have epigenomics, which seeks for the modification of DNA that not affect this sequence. But why does this all matter? Well, studying all these levels help us understand exactly how things work in living things, and we then can explain the phenotype of a specific biological system in a more holistic way. So back in the past, not in the past, but back in the day, scientists used to look at just one thing at a time, like a single gene or a single protein. But omic sciences are different. They check out all the molecules from the same biological type, giving us a way bigger picture. And what is more, we can take all these layers of information, mix them together, and connect the dots, and come up with some plausible conclusions that, in our case, we use to figure out why the Mediterranean diet is so good for our health and which mechanisms are promoting these effects. So what are the omic strategies that we apply in this project? We employed metabolomics through NMR and also through mass spectrometry, and metagenomics through massive sequencing, all of them to scrutinize the gut environment. So the objective was to discern this interplay between metabolic processes and the composition of the fecal microbiota in this context of dietary intervention. So since the gastrointestinal tract is or has a pivotal role in organism homeostasis, because it's involved in nutrient absorption and also it performs complex fermentative activities, the application of these metabolomics and metagenomics allow us to to the picker this interaction in this dynamic ecosystem. So to summary, in that case, metabolomics analyzed the small molecules within the gut, shedding light on the metabolic consequence of the dietary intervention. And in a similar way, the metagenomic approach elucidated changes in taxonomic composition of the gut microbiota in this context of dietary intervention. So now let's move to the juicy part of the project. That is what we found in the MED4Youth project using these omics sciences. One of the remarkable thing of analyzing omics sciences is that when we analyze an omic data set, is the ability to treat all the variables as a comprehensive set, not the variables as a single element, but as a comprehensive set. This set can then be used to discern differences among experimental groups by plotting them in a PCR plot. Here you can see a PCR plot on the left side of the screen. In that kind of plots, the metabolite set for each participant is displayed and categorized in the respective dietary interventions. And unfortunately, we not see clear separation in that case. So we expected to see differentiated clusters. So this suggests that the overall metabolite set in a stool do not significantly differ among the two dietary interventions. Nevertheless, from this within this broader metabolite set, we can analyze individually specific metabolites. And in fact, this is what we do it. And we found significant variations in lysine and in fumarate levels among the different dietary interventions. Changes in lysine may be associated with protein intake from meat, eggs, fish, dairy, and also can be related to protein absorption. And on the other hand, fumarate is an intermediate product in certain microbial metabolic pathways, suggesting an active involvement of the microbial composition. And fumarate is also present in vegetables, which can link with the results that Alice presented before, showing an increased plant-based food intake in Mediterranean, in the participants that were adhered to Mediterranean diet. And furthermore, we observed an increase in these three metabolites, but only in the low-fat diet group. The first two metabolites are directly linked to microbial activity, while the presence of glucose in stool suggests alterations in glycolysis. Now, shifting our focus to the bacterial composition of the intestinal microbiota, again, in this PCR plot, we noticed a 10 of the microbial communities to cluster based on the dietary group. This clustering is not significant, but the centroids are visibly separated, which means that at least few differences in bacterial compositions are present. In fact, we identified taxons that exhibit significant changes corresponding to the dietary intervention. Clostridium and Lachnoclostridium demonstrated a tendency to decrease in the Mediterranean diet group, while Probotella remains without change. I mean, it didn't change in the Mediterranean group, but it increased in the low-fat diet groups. And of particular interest, Lachnoclostridium has recently been found to have a positive correlation with visceral fat, which can also make sense in our project. And after completing the individual analysis of the omic data sets, our focus shifted towards exploring a correlation among various data sets generated in this project. These include variables from different sets, being gut-derived metabolites, biochemical variables, which are clinical, anthropometrical, and food intake biomarkers, and also this bacterial taxonomy. So what we did was to specifically select these data sets and try to correlate and integrate them using Diablo from Mixomics R package. First, we correlate the variables to construct a correlation table and a circle plot. On the lower side of the I don't know the word in English, of the diagonal, you can find the correlation values between the data sets 2 by 2. For instance, between the metabolites obtained by NMR and the bacterial composition, there is a correlation of 0.62. In fact, this is the strongest correlation observed between data sets. And a graphical representation of this can be shown in this cycle plot, where once again we observe that data sets with the strongest correlation are those of metabolites, good derived metabolites, and bacterial genera. While the correlation may not be notably strong, noteworthy patterns emerge. For instance, we can see here in this Diablo graph, I think I will try to show. Can you see my? I think in the circumference represent the selected variables from each data set. Okay, these lines connecting them depict the correlations with a cutoff of 0.7. The orange color means positive correlations, and the black color represent the negative ones. And these lines that we can see here around the circumference display the expression levels of each variable, and also separated by experimental group. And for example, here we can see that fructose negatively correlate with valeric acid, and both components exhibit differences in their levels depending on the type of diet. More specifically, I will not enter into detail, but we have seen that certain bacterial genera show both positive and negative correlation with several metabolites, and suggest this interplay between these two layers of biological information. After that, we represent each participant in a PCA plot considering the unique combination of metabolites, bacterial genera, and biochemical data. So when we consider this combination of this information and try to represent this by a single dot, we try to assess whether this combination reveals a clustering that can separate the participants depending on the dietary intervention. This, as you can see, this PCA plot does not really exhibit a clear clustering pattern. We would expect something more like this, with separate clusters. So our results indicate that the dietary effects of both interventions are quite similar and don't influence these variables in a strong different way. So to conclude, based on omic analysis, we did not see clear differentiation according to the type of diet. Several factors could contribute to this observation. Firstly, the small sample size. The recruitment was difficult during the pandemic, and this may limit the statistical power. Also, it is plausible that the differences between the two diets are not substantial enough to induce these molecular level changes. And lastly, we also have to consider that poor adherence to the diets could explain this absence of molecular distinction between them. But in light of these results, we have the proposition to explore the effect of the dietary treatment on health without distinguishing between diet types. And this approach aims to correlate this omic data with the observed anthropometric effects, and these analyses are currently underway. And last but not least, I cannot end without saying that we confirm that integration and correlation of data help in interpreting a biological system. And although more detailed investigation are scheduled for the coming months, we can say that this approach is useful in the dietary intervention. So that's it. I'd like to thank all the researchers involved in the project, and also thank you for your attention.
Okay, thank you very much, Elena. I think you have answered with the last slide one of my questions that I wanted to ask you regarding indeed if you were expecting something different, and if not, why we don't think it hasn't worked as maybe expected. But please, maybe I will now kindly ask all the previous speakers to switch on cameras, and we will take some of the questions now. And please, all the attendees, if you have additional, post it in the chat. We had a very previous one from the beginning regarding the meal plan and the educational material. Here, the thing with MED4Youth is that the material was specifically designed for the children, so it was more or less, Licia, you can correct me the wording, but it was more like personalized. But similar work more for general population is done within the Switch to Healthy project, and I think here the material, if not all the material, at least I think quite a bunch of material will be published, Simona, if I'm not wrong.
Yes,
and,
I confirm.
So you can follow also now Switch to Healthy, and you can, I guess, if not at the end of the project, in the following month, you can get information regarding the educational materials and meal plans. I have, let's see, starting, I have one question for you, Simona, or maybe I would say a couple of questions. One is, because I think this is quite a big difference between Switch to Healthy and MED4Youth, that you had this starting co-creation as a step phase in which indeed is engaging the future user from the beginning into the research. And maybe if you can give us some insight of the barriers or the feedback that you got from these co-creations, regarding maybe changing habits, if they see easy or not, or if there is social, economic, other barriers that are hidden behind this.
Yes, indeed. We started since the very beginning with, first of all, drafting the concept together with the end user, in a sense that through our partners, we already selected some of the needs of the specific target groups, so since the very beginning. And then we kicked our phase with a very huge survey campaign in order to get insight from different countries all across the Mediterranean area. So we got involved Lebanon, Egypt, Morocco, Turkey, Italy, and Spain. And this was one of the key factors that help us to shape the next steps. So we collected more than, as said, 1,000 responses in our huge surveys, and from them we shaped the focus group actually in the 3 target countries, so Spain, Morocco, and Turkey. And specifically, we ran different focus groups. First of all, up to know their current habits, and secondly, really to select what food prototypes they would prefer the most, or which features in IT2 would be helpful for them to really to ensure us that they will actually use those tools. And of course, the key differences is in the different countries, so the different habits across the countries. So we have 3 representative, one from the more Arab side, Turkey, which is right in the middle between the European and the Arabian habits and behavior, which is in a way most similar to Greek in terms of habits. And we have then Spain that is another completely different approach. So after we have this very clear picture, we are ensure that we really have the key elements to design in a very tailored and specific way the intervention studies, so all the solution that we are going to have. So for example, and this is something that CERT is going to give you a detail also in the next session, is really to customize also the meal plans according to their habits. So we ensure that although we want to promote the local biodiversity, introducing, some kind of neglected products or ingredients, also to be sure that they are healthy, of course, but they follow in a way their current habits. So really the behavioral change will be made step by step.
Mm-hmm. Thank you, Simona. And then another, I would say, more generalistic that I think it would go also for Simona or also for my colleague Antoni, is that okay? Now we have the results, no, but we know it's about how we can really create this environment and how we can really bring these results into creating impact, or moving forward with these results. So I don't know, maybe in Switch to Healthy, how is the pathway or the approach towards then use the results and disseminate and make really the most of them.
Yes, I will go first. So yes, as we said, as I said, we are going to create an enabling environment, and this means that we first have to help business, so enterprises, to have concrete roadmaps to put the products, the novel products and the prototypes into the market. Secondly, we have this huge engagement with the schools, families. So we are target really people in this sense. And this is something that I really trust that will make the difference, because they feel they are belonging to the solution. They are part of the solution because they are co-creator of the solutions. And we engage with the 3 governmental authority, 2 regional and 1 national, from Italy, from Spain, from Catalonia specifically, and also the Ministry of Tourism. For us would be the ambassadors for the Mediterranean diet. So why we engage them? In order to really, as said, create from the regulatory point of view, from the policy point of view, an environment that helps all these stakeholders really to have a way to back to the tradition. So in this sense, they are going to develop new policy instrument together with us, really to valorize the results. So it is not only a research and innovation action, but really we want to have this also from the policy perspective change, because we have policymakers them, so they will help us to shape it, to raise the voice. So we are not going to make any lobby on them and advocacy, because they are part of these groups. So for sure, they will be very supportive for the future.
Okay, thanks, Simona. I don't know, Tony, if you want to add, because also in the case of MED4Youth, we have also a very key role of the nutritionist, in here. So it's also another stakeholder, let's see, in this value chain.
Yeah, in my opinion, there are different keys to have success in preventing or tackling youth obesity. One is to have the motivation of the parents to boost the healthy habits to their children. No, this is key, and we have seen in our study that when the parents were more compromised, the results were better. And for me, this is very important, to implement the healthy lifestyles at home, and to have the engagement of the parents in this very sensitive population. This is on one hand for me is key. In addition to convince that the adolescent, this is a work that needs to be done by the parents. That the addition, the physicians can help them, but it is very important that they trust them and they convince their children. This is for me very important. And the other, for me, key point is also the policymakers and the politicians, okay? Because it is important to maybe to include policies to penalize the consumption of unhealthy foods with taxes. This is important as well, no? And this is there were some attempts to do that, but maybe this could be implemented, because maybe sometimes the unhealthier foods are cheaper than in most cases, no? And this is not a good thing, no? To promote healthy habits, no? And of course, also the companies focus on Mediterranean diet-based foods, and also have a very important role, not to promote the Mediterranean diet, because the adherence to the Mediterranean diet among the young people is lower than 20% in Mediterranean countries, and this is very surprising, and it's a pity, taking into account the well-known reported healthy effects on the Mediterranean diet, and I think that we need to boost the adherence to the Mediterranean diet.
Thank you both. Yeah, indeed, it's research plus industry, and then policy making in general. So you also both pointed, then we'll see in the next session the relevant role of the food industry, and we'll have here indeed 2 examples that we'll be presenting. So now let me take a more research-based, I would say, question that will start from the beginning. One question regarding how would you link the fact that the low-fat diet produces an increase in the circulating levels of glucose and a decrease in triglycerides? Are the levels of both parameters within the healthy range? And I think this was more for, Alice or Paolo. Paolo. Yeah. The next one, yes, it's for Richard. Yeah.
Well, I cannot say too much about that, because I think we don't have a conclusive explanation for that. But in my personal point of view, it may be related with the fact that we have a fat-restricted diet, and this may lead to a reduction in fatty acids and then in triglycerides, but we didn't measure free fatty acids. So the conclusive answer for this, it would be, in my opinion, to measure the free fatty acids in the plasma. And we may have a compensatory increase in glucose, and in particular, this may be important because this is not yet adult population, and the metabolism may be different from the adults, in which probably a reduction of the fat would not lead to an increase of glucose. And here we may have a different response to the diet. Although the range of the glucose levels was within the normal range. It was not like an hyperglycemia. It's not the case. It was a statistical significance. It may not have any clinical significance. It's a completely different dimension of data analysis. We sometimes have clinical differences that do not have a statistical significance, and have statistical differences that do not have clinical significance. So this should be analyzed from these 2 different perspectives, and we have to think about. Yeah.
Okay, thank you, Paolo. Now I think to Elena. No question regarding the scoring for the variables, the taxonomy, the biomedical, the metabolites. How we generate this kind of scoring, the zeta score for each component, and the sum of all those components?
Well, this is a tough one. I don't know exactly the math behind this, but we use Diablo to perform this analysis. And as far as I know, for each variable, we get a contribution score that is like a loading value that gives us a notion of the importance of this variable to explain the group, that in this case is the type of diet. And then we applied different models, including PLS and SPLS, that are based on partial least square for variable selection, and then apply this model for a combination of all the data sets. So in the last PCA that I show that I shown that all the data sets were mixed, the numbers that were represented were that the contribution of each data set is average in a weighted way, because some data sets explain more than the other one. And then this loading score represent this mean loading score is represented. So I don't know if I'm answering the question, because it's difficult for me to explain this, but the basic thing is that we are trying to get a number that explains the contribution of this variable to explain our experimental groups. So we are trying to quantify the contribution of each variable and then represent it in a PCR plot.
Thank you, Elena. And I said also before, if no further information is you wanted to get in touch with any of the speakers, will give you also an email address. So now let me, I would say one or two more, but I think now it goes to Alice, and then we move on with the webinar. Regarding the meal plan, no, we recommended 3 portions of legumes per week, and we're asking why not more?
Okay, so the picture in which you saw the 3 portion of the weekly recommendation was related only to the products we provided volunteers, so only the chickpeas. So in our prescription, we suggest to have at least 3 portion of legumes per week, not just 3, but at least 3. And of course, we base our prescription on the food-based and dietary guidelines. So that suggested for the youth population having at least 3 portions of legumes. So this is why. Even you have also to consider that the majority of participants at the beginning of the study did not eat any legumes at all. So it's better to change a little the diet of participants step by step. So reaching at least the suggesting portions and then try to increase in the future, maybe, because otherwise we will know that the volunteer will not follow our dietary prescription.
Okay, thank you. And I take the next one, and I will leave the final word for maybe next block. So I think also I see a key one as well, because at least in the MED4Youth, if we have not maybe see such a huge difference between the Mediterranean diet intervention and the low fat diet regarding these children and adolescents obesity, and also we are always thinking on the follow up after the intervention, because maybe once we are in interventions, people might be more prone to follow, but then it's about maintaining these habits or the change of habits. So we can think or elaborate maybe on the fact that Mediterranean diet might be more palatable diet, and this might long term help to increase adherence, maybe regarding the low fat diet might be this way, or what do you think, Alice?
Yes, of course. But first of all, I want to say that not reaching any significant differences in the dietary treatment for us, it's a good result, because at least we were able to demonstrate that the Mediterranean diet was not less effective than the traditional clinical treatment for youth obesity. So we can suggest that in specific countries, like the countries of the Mediterranean basins, we can use alternative treatment like the Mediterranean diet, because it's easier. Probably it's easier for the adolescents, but also for the whole family, because at the end, when we talk about youth, we have to consider all the family. Having a Mediterranean diet that it's more linked to their preferences, their habits, and the cultural and enogastronomic heritage. So of course, probably it's easier to follow also at the end of the intervention. And we also have to consider that the Mediterranean diet has a lot of other aspects other than nutrition. So, for example, the social cultural aspect, the physical activity, and all. It's called a lifestyle, a Mediterranean lifestyle, and not only the diet. So this is very important. Also considering the environmental aspects that are very crucial in this period. And the Mediterranean diet was also suggested as environmental, as a diet with a lower environmental impact than, for example, other dietary models. So I think it could help in the future also for after the intervention itself.
Thank you, Alice. So let's see, yeah, maybe the last one. So we maybe close the block regarding if you've seen any relationship between Mediterranean diet and sleep habits in relation to obesity and overweight. Tori, maybe?
I can answer it, but it is a very good question, because I talked maybe 1 year ago with Professor Ordovas, and he suggested me to check this, because it has been seen that obesity can be linked to a nocturnal pattern more than a diurnal pattern. But unfortunately, in our study, we didn't check this in deep. But it has been it is a very good point to check in further studies, because the relationship between obesity and circadian rhythms and sleep patterns is known and is explored in more detail in current studies nowadays. Okay, but a very good point.
Thank you, Tony. So I think, yeah, we move. Thank you very much, all the speakers from this first session. We will now shift to, let's see, the second building block that also share between the two projects, where there is a key role from the industry. And we'll start with Professor Guillermo Saldana from Novapan. Yeah, Guillermo, he is the head of the R&D department in the bakery Novapan SL. He holds a degree in veterinary science, food science and technology, and PhD in new technologies of food processing science. And he is a scientist and a food enthusiast with experience in food microbiology, food preservation, new food processing technologies, and development of new food products. And please, Guillermo, you will talk about Novapan products in MED4Youth.
Okay, thank you very much for the presentation. Not yet professor, but thank you for the upgrade. Good, so let's go for it. Okay, so the idea is to just give some tips about the bread, because It is a very traditional and basic food in the Mediterranean diet, and there are still some questions for the consumers. Let me introduce our company, our brand. We are more known for our brand PANISHOP. It is a familiar company that have been involved in the bakery industry since the very beginning of the 20th century. This company have been growing until reaching 40 shops all around Spain, mainly in Zaragoza, but also with presence in Madrid, Huesca, and Tarragona and Reus. We are also serving to some supermarkets like Mercadona or Lidl, which is more international. We are also known for using sourdough for the fermentation of our breads. Ours is one of the best sourdoughs recognized. That was the first sourdough included in the sourdough library in San Vit. And also this company is putting a lot of effort in research and development, and we have been involved in more than 4 or 5 research projects until now. One of those projects is the MED4Youth project that has been already presented in this season. So the objective of PANISHOP within the MED4Youth project would be something like developing a sourdough bread with proven healthy properties as part of the Mediterranean diet. So the first question that should come to our head is bread a healthy food? Well, the short answer would be yes, it is. But we are going more into detail, otherwise the presentation would finish here and would be a little bit short, right? It is a healthy food. Bread as a source of carbohydrates is in the base of the nutritional pyramid or the Harvard plate. It has low fats and sugars, and the front indicators like Nutri-Score are always giving the best rates for most types of the bread. It is a food rich in complex carbohydrates. The World Health Organization is recommending to obtain 50% of the energy from the diet coming from carbohydrates. It also provides fiber, regardless if it's a white bread or whole grain bread. Of course, it is a better source of fiber for whole grain breads. Other benefits coming from whole grains, not only from the fiber. It improves the gut function, the growing of positive microorganisms, reduces the risk of obesity, diabetes type 2, and also a protective effect against cardiovascular diseases. When the bread is fermented with sourdough, let's say sourdough bread, it also the glycemic index is reduced. It is a more digestible food, and sometimes we can also find bioactive components after the sourdough fermentation, and it's also always good for the positive gut microbiome. It is a simple food. Bread is something really simple, made with flour, salt, water, and ferment. Basically, it can be also more complex with different flours or seeds, but it's made only with those for ingredients. The carbohydrates coming from flour, as I said, are providing large number of the energy that we should consume during the day. The fiber is the food for the gut microbiota, for the positive food microbiota, like for example lactobacilli or bifidobacteria, and also improves the gut barrier. The fiber is coming from whole grain cereals and the whole grain silver cereals or seeds in general. Also, the whole grain cereals and silver cereals like buckwheat, for example, are providing polyphenols, which are antioxidants and also inhibit the growth of pathogens in the intestine. And also the polyunsaturated fatty acids coming again from whole grain cereals and seeds are improving the adhesion of those bacteria to gut mucosa, but also having an important cardiovascular protective effect in the consumer. Talking about sourdough, which is something for us very important, let's put a little bit of context here. The Spanish regulation is, well, I would say new. It's from 2019, but that's one of the first regulations putting on the table what should be a sourdough bread and a definition of sourdough. It says that a sourdough must be an alive culture of lactic acid bacteria and wild yeasts, and those microorganisms must come from an spontaneous come from a starter. It also has a definition for inactive sourdough, which is the sourdough that has been inactivated by some treatments and therefore cannot ferment, cannot proof the dough. The sourdough bread must contain at least 5% of sourdough, and that's also something important. The use of additives is not permitted. The use of baker's yeast, meaning Saccharomyces cerevisiae, inoculated Saccharomyces cerevisiae, can only be present in a very, very low concentration. It also has a definition for for some control parameters for the sourdough, like pH and total treatable acidity. And the same for the pH of the bread, putting a maximum level of pH 4.8 for the baked bread, meaning that the consumer can be sure that this bread has been fermented with an acidic dough, a real sourdough, and then having all the benefits of this fermentation in the product that they are consuming. Therefore, this law is mainly written to protect the interests and the health of the final consumer. Although I said that the presence of Saccharomyces cerevisiae cannot be present in more than 0.2%, in that case, I was referring to the inoculated, to the baker's yeast. Nevertheless, Saccharomyces cerevisiae is one of the most present yeast in general in sourdoughs, although other yeasts are present, like for example Cathastania humilis or Cathastania exigua. More than 100 yeasts have been identified in different sourdough samples all around the world in the scientific literature. Regarding the lactic acid bacteria, the most common lactic bacteria are coming from the genus Lactobacillus, mainly Plantarum brevis or San franciscensis, but also other bacteria are present. More than several hundreds of lactic acid bacteria have been identified in sourdough samples. This mixture of yeast and bacteria are having an effect on the dough fermentation. They are consuming sugars to producing organic acids, meaning that the glycemic index of this bread is lowered, and that's always something positive. But also because of the action of these organic acids, the shelf life of the bread can be extended because organic acids are natural preservatives. Well, this acidic environment, together with the enzymes coming from the cereals, but also the enzymes coming from the microorganisms, are pre-digesting the proteins, mainly the gluten. Gluten is not toxic itself, but can be difficult to digest by the human body, mainly when there is a previous disease in the consumer. Therefore, this pre-digestion during the sourdough fermentation make more digestible breads, but also has a technical positive effects like bread more soft, and also producing flavors and colors typical for slow and sourdough fermentations. It has been described that some lactic acid bacteria can produce some kind of fibers, exopolysaccharides. Those fibers are considered prebiotics, and therefore are improving the profile of the gut microbiota. But also those fibers are retaining water in the bread and in the final bread, meaning better bread volume and softer texture of the bread fermented with sourdough. In this table, there is well, the same explanations that I just given, so I just skip this slide. We have been involved in research project since 2017 with the previous project Pansalud. In this bread, in this project, the idea was to develop different breads with different sourdoughs, then feed rats with laboratory rats with those breads, and see how they change their health status. What we just in a short summary, we saw that the sourdough breads made with spelt wheat, durum wheat, and multigrain showed lower blood glucose than other breads, and especially sourdough multigrain bread showed a lower inflammatory reaction in the intestine of those rats, meaning that was more digestible. That's why from this project, the best combination was the bread made with sourdough and a combination of different cereals and grains. Coming from the MED4Youth project, where first question was how to make more attractive bread for the consumers, because you can produce something very healthy, but if the consumer is not accepting it, then it makes no effect. So in the 3 different countries taking place in this project, Spain, Portugal, and Italy, the results were more or less similar. The most appreciated kind of bread was the white bread, but also the soft texture and crispy crust were also attributes that were well appreciated by the teenagers from those 3 countries. So with this with this idea in mind, we developed a sandwich multigrain bread, which was based on the multigrain Pansalud bread, and also baguette, including byproducts from the cider industry, meaning the apple pomace and also the cider yeasts. Sorry. Taking in consideration the analytics from the MED4Youth project and the nutritional profile, and also the consumption survey, again, the multigrain sourdough bread from Pansalud was the best candidate for including in the nutritional intervention of the MED4Youth project. But well, those other two breads were also interesting, but multigrain was the winner. For example, taking a look in the nutritional profile, the multigrain bread showed a very low energy content, very high fiber content, and a low sugars concentration. So those were aspects very positive for including this bread in the nutritional intervention of the Mediterranean diet of the MED4Youth project. As a result of those projects, we are happy to see that we publish those results in the scientific paper that is on the screen. For example, just an example of the results showing how the multigrain bread with sourdough showed lower glucose concentration in blood after postprandial consumption. So that was a very, very short intervention. Just let me tell you thank you for the opportunity to dignify a little bit this basic food that is the bread present in all the cultures, in all the history all around the world. I hope that the question that if the bread is a healthy food now is clear for everyone that yes, it is a healthy food. And again, thank you very much for your attention to this short presentation.
Thank you, Guillermo. So let's take the next presentation, and then I think a couple of nice questions regarding this block of food products. So yes, next speaker is Sonia Sanchez. She's great in agricultural engineering at the University Rovira i Virgili. She has been working at Della for almost 7 years as a product developer in R&D. Sonia, I think she's going to speak about the healthy snacks developing within Switch to Healthy project.
Okay, thanks for introducing me. Okay, good morning, everyone. I'm going to share the screen. Okay, let me see. Can you see it properly?
Yes, Sonia.
Okay. Okay, then. So, morning, everyone. Well, Simona just explained perfectly well the Switch to Healthy project. So now I'm going to get more focus on the prototypes, okay? And I want to start saying that as a developer, I have the commitment, let's say, of create an eco-design product. And it means that we have the compromise not only to develop tasty and healthy products, but also with ingredients nearby, the most nutritive ones from our farmers. And okay, and of course, we are going to use a ready-to-go packaging for these products, because everybody nowadays is in a rush. Everybody wants the things ready to go. So, and this has to be a monomaterial to be recyclable, and once we get nourished, just throw it away and be careful with the environment as well. So well, the aim of the project, as Simona said, is generate an eating behavior change towards the Mediterranean dietary pattern, and therefore to a healthy way of living. We think about that, no? As I said before, let's start to do this with ready-to-go products to replace the less healthy snack options that we have in supermarkets and in other places. And for that, we are going to get focused and to think about those key ingredients, the ones from our Mediterranean area that we're talking about the whole morning. So as the seeds, for example, we are going to use quinoa and flax seeds, cereals, always whole grain cereals like rice and oats, legumes as well. We are going to use some in some prototypes as chickpeas, of course, fruits and veggies. As a veggie, for example, butternut and sweet potatoes, and as fruit, prunes, peaches, bananas, apples, and mandarins. And now it's time for the mandarins, so mandarins as well. And nuts, we're going to use almond and hazelnuts, and because of the good fatty acids, as it's well known. And here we are going to talk about, we are going to have 8 prototypes, okay? And I'm going to divide that in 5. It's going to be done in a pouch format, okay? And the other 3 in cups. Okay, so here we have some of the prototypes, and we're going to use yogurt as a Greek yogurt, another ingredients are sumac, fiber, rice, quinoa, almond, the ones I told and I told to you before. And here the important thing is that we are going to do as well a nutritious product. And here there are some of the claims that we are going to try to achieve. For example, high content in fiber, some vitamins of C and vitamin C, omega-3 because of the oils, and also vitamin E. Those are the ones in pouch, and in cups we are going to have as well fiber, protein, and those are the main claims we're going to achieve. Of course, you know that nuts and seeds, those has, as I said before, good fatty acids that helps with to be nutritious, those products, and always thinking as well about the texture, because those products are going to be for kids, and of course they are going to they could be taken in for adults, but the focus, the main group is kids, and that's why it has to be attractive as well, not just because of the taste, but also of the thickness and the ingredients, the sweetness. So we have those 8 prototypes, and after an external partner test, we're only going to be focusing 4 prototypes, the ones we are going to be chosen, and those are the ones we are going to carry out with the studies for the families here in Spain, and well, we think that they are going to be a good start for let's say more, get the people more focused or more with this Mediterranean diet, that it's so important what we are talking about the whole day. So let's see if we can achieve this objective. Okay, so now my presentation. And thank you so much for your attention.
Thank you, Sonia. We have time maybe to have one question maybe for each of you, and then I will start with Guillermo. I think it's crystal clear, no, that bread yes or bread no is a yes. But what about replacing bread by other carbohydrate sources in the diet?
Well, this question should be carefully answered. First question, first would be, why to replacing bread? Is there any previous pathology, like for example, celiac disease or any intolerance? Then, if this is the case, of course, gluten should be avoided. In case there is no previous pathology in the consumer, we also have to consider the positive aspects of any other sources of carbohydrates. Remember that bread is not only providing carbohydrates, but also fiber and other nutrients like polyunsaturated fatty acids, and so. And finally, the bread is a fermented food with this fermentation process is always positive for the nutritional aspects. So changing bread as source of carbohydrates by other source of carbohydrates which is not fermented raw, I wouldn't say that other foods are negative for health, but bread could be more positive than other carbohydrate sources.
Thank you, Guillermo. I think it's clear now. Keep on no, if no, we should keep on no with bread as it was no previously, no more tradition in our tables. And now one for Sonia, it was more regarding the plant-based snacks, no, that are you going to develop within the Switch to Healthy. We know that we really need to move to sustainability, but do you have any insights of how the consumers might react to those type of products or those ingredients? I would say more from the local biodiversity, but also from more a traditional point of view. Do you expect that open to uptake them, or there might be still a little bit of reluctancy towards these kind of plant-based snacks?
Yeah, well, we think that we are using a very common ingredient, so people is going to be very familiar with it, with the taste, with the texture. So I think we are not going to have, let's say, problems to those prototypes, and everything is going to be okay with that. Yeah.
Okay. So thank you both, Guillermo and Sonia, for explaining the perspective for the products that you are developing you have developed in the different projects. And finally, we'll go straight to the final session. The last but not least, also share among the two projects, the MED4Youth and the Switch to Healthy, which is the use a little bit of digital support. We are in the era of digitalization, and then it's also now used to support a little bit engagement and sustained dietary patterns. So first speaker is Perla De Innocenti. She's currently second year at PhD student at the Food Science Human Nutrition Department of Food and Drugs at the University. Bachelor's degree in Biological Sciences at the University of Padova, master's degree in Human Nutrition Sciences at the University of Parma, and research fellowship at the University of Parma as well. So, Perla, the floor is yours.
Hi, Beata. Thank you for your kind introduction. I'll share my screen. Are you able to see correctly the presentation? Okay, perfect.
Yeah.
So it's a pleasure to be here today and talk with you about the MED4Youth application, especially concerning the contents and the usability of this app for the adolescents and their parents. So I will discuss about the structure of this app, its usability, and then the final considerations. This web application was created with two main purposes. First of all, collecting data, as you already seen in the previous presentation of my colleague Alice. Among them, the physical activity level, the adherence to the Mediterranean diet, and, for example, quality of life. Second aim, no less important, improve the adherence to the Mediterranean diet and lifestyle behaviors of participants, using a learning through playing approach. So, in other words, not only material to be read, but also quizzes and trophies. In addition, consider that the app had two separate accounts, one for the adolescent user and the other for the parent. And this was helpful to obtain more targeted contents according to the age range. Of course, before starting the intervention, we perform a usability trial involving 12 subjects, and we ask them to fill out a 10-item questionnaire about general information and also about the app. And below you had some example. For example, on the left, you can see that the majority didn't use a similar app before. And on the right, they thought that this new app could be easy to use, but also interesting and useful. In addition, we asked them to test all the functions of the application. And from that type of feedback, we decided to add some additional buttons and also to use a common food database that was the SQL French database. But now, moving more in detail about the structure and the functionalities of this web app, consider that it was a cross-browser web application, available not only for the computer, but also for mobile devices. And the user could simply enter in the login page, insert its username and specific password that were provided at the beginning of the intervention, and then open the home page. As you can see below, there were different sections according to the different contents. Always talking about the structure, here you have an overview of the different pages inside the app. For the adolescent, you have the dots in yellow, while for the parents, the contents are represented with the dots in blue. And all the app and the contents were translated also in Italian, Portuguese, and Spanish. Focusing now on the intervention phase, consider that for the adolescent user, we set a short period of time before each visit to compile the food diary and also the questionnaires, while for the educational contents, they were available for all the intervention period. And the adolescent could check his weekly meal plan, but also a new topic every week that was also associated with tips and quizzes. And in general, we provided also some trophies. While the parents user could check also the same weekly meal plan of their children and also had the same topics, but explain it in a different way, and in addition, recipes. This is important because the app was the same for both intervention group. The main difference was only in the weekly meal plan that depended on the intervention group, of course. Now, moving in detail about the educational content of the adolescent user. As you already seen in the presentation of Alice, this is a scheme that was provided only to the intervention, the Mediterranean group, to explain to the volunteers how to include the study products inside the weekly meal plan. And this is the plan, always for the Mediterranean group, but for the structure, it was very similar to the low-fat diet. And important to specify that we provide them only one meal plan for all the intervention period, with the possibility to change the foods inside each food group using the icons. So, for example, in the case of fruit, we had the icon of the apple. Here you have two screenshots, one for the questionnaires and the other for the food diary. There is the example of the KidMed questionnaire, and you can see that it was very simple to selecting one of the options available inside the questionnaire. While for the food diary, we gave us two possibilities. So first one, insert the food, selecting the food inside the list of the database and also specifying the quantity in grams. But also they could simply typing in a free way the name of the food, also specifying, for example, the name of the brand. And also for the quantity, they could specifying one slice of lasagna, for example, and 1 cup of tea. Now, about the topics and the tips, you have an example here of the topic of nutrients. As you can see, it long respect to the tips that were more shorter. And always about the topics, in total they were 17, and only the second one was a little bit different according to the intervention group. Again, about quizzes and trophies, here you have two examples of screenshots. And for the trophies, they could be one, simply checking, for example, the topics, the tips, but also compiling the food diaries and the questionnaires. But finally, we can discuss about the usability of this web app. And we can say that in general, participants from both groups use the app mainly to consult the food plan. And especially in Italy, they use the app to compile the questionnaires and the food diaries. Sometimes we provided also the paper format. While for the other contents, they use the app less frequently. But in addition, we asked to the participant to provide us a feedback always about the usability. And from the adolescents, we understood that the app wasn't so very practical. They would prefer a mobile app, for example, with notifications inside to be able to remember when check the different contents and also when compiling the diaries and the questionnaires. And for someone, the diaries sometimes were difficult to compile using the database inside the app. But in general, for the one that use the app, they like the tips, topics, and also the games available. Interesting also the part of the social component, because someone asked us also to add this part in order to interact with other users inside the intervention study. While for parents, they use the app to check the different recipes, and they ask us also to add more recipes each day, and also to add the possibility to download these recipes as a PDF file. Others use the app to check mainly the diets. And for the one in general that use the app, they thought that it was very interesting learning more about healthy eating. But for the other that didn't use the app a lot, they told us that it was mainly because of time reason. So about final considerations, we can say that the app had, of course, positive aspects. So it was helpful to collect data and also homogenize that part for all the 3 countries. In addition, could be an efficient system to easily interact with people, especially the young. But of course, there are several things that need to be improved. For example, change the web application into a mobile one, but also add inside a more interactive system for the user with, for example, the notification and the messages, and of course, the part of the social component for the interaction with other users. And not less important, maybe simplifying also the food diary, for example, adding more commercial food and with standard quantities available. Well, that's all. So special thanks to the whole amazing team. It was a pleasure being part of that research group. And thank you all for following.
Thank you very much, Perla. Next speaker is Lazaros Gymnopoulos, that he is going to talk about the Switch to Healthy app. Lazaros, he is a research assistant at the Information Technologies Institute of the Center for Research and Technology, ELAS, the third. He holds a diploma in electrical and computer engineering from the Aristotle University of Thessaloniki and an MS in information system from the Athens University of Economics and Businesses. Please, Lazaros.
Thank you, Bioza, for the introduction and the invitation. So let me share my screen. Can you see my screen?
Yes, now we see.
Great. Good afternoon, everyone. My name is Lazaros Gymnopoulos. I am a research assistant at the Center for Research and Technology, ELAS, and I'm managing the Switch to Healthy project on behalf of CERT. Today, I will present the Switch to Healthy application. And if we have some time, maybe we can say a few things about a game we are also developing. The Switch to Healthy application is aimed to be an AI-powered, web-based, personalized nutrition application. AI-powered in the sense that it employs an artificial intelligence algorithm for the dietary recommendations. It is web-based, so it can be accessed via any common browser, and therefore any device that supports a browser. And it provides personalized meal plans which follow the principles of the Mediterranean diet. The personalization is based on the user profile and specifically on physical characteristics of the user, his PAL, allergies, dietary preferences, and preferred cuisine. 3 cuisines are supported: Spanish, Turkish, and Moroccan will be added because these were the partners that we are cooperating within the project. It supports a MED-based diet, and this is ensured via a set of rules defined by nutrition experts. So I have some screenshots here of these rules. It is targeted mainly at the EU citizen, but it is also good for an education or a health professional. And also the application at the end of the project will integrate educational material. Now, the current status of the application. Currently, we are developing 3 separate applications. One that covers the functionality for the adults, one, and it can be accessed also in this link. One that covers functionality for children, and also the educational game that I mentioned. The 3 separate applications are currently under beta testing within the project, and at the end of the project, they will be integrated into a single application aimed at whole families. This is planned for around May 2024. Today, we will present the application for the functionality for adults. Also, I forgot to mention that the game and the first application are developed by CERT, and the functionality for children from a project partner, the University of Parma. So a few technical information. The front end of the application was designed through a design sprint procedure, where a group of people designed different mock-ups for each possible screen, collaborated to select the actual screens of the application, and voted to select the best mock-ups to be used for the application design. And it was implemented in React.js. The backend of the application is where we implement the AI recommendation system. It is implemented in Django, and an appropriate database schema was designed initially, and the corresponding DB was implemented to store all the necessary information. Now, the user profile. These are the elements of the user profile. As you can see here, that we have personal information, physical characteristics, dietary choices, and some allergies, and the selection of the cuisine. Now, the food database was created by experts within the project, by dietitians and nutritionists that are partners in the project, and it is comprised of dishes and meals. Meals contain 1 or more dishes. Meal combinations are called nutrition plans, and the daily nutrition plan consists of exactly 5 meals: breakfast, morning snack, lunch, afternoon snack, and dinner. A weekly nutrition plan consists, of course, of 7 daily NPs. All necessary information for the calculation of an appropriate plan and a healthy plan is contained at the dish level, so that we can propagate all these properties to all other levels. Here you can see a small part of the database that was created by the project experts, and we thank them very much for their contribution. And you can see that there are a lot of details for each specific dish. Now, a few words about the algorithm. Initially, we filter the meals for each user profile, considering, for example, user allergies or dietary choices. The next step is the generation of many candidate daily NPs. So all possible combinations of the filtered meals are taken into account. And the only necessary attribute at this point is that they contain exactly 1 meal of its type: breakfast, lunch, etc. The next step is to select the 7 most appropriate daily nutrition plans for the specific user. So this happens with first scoring each daily nutrition plan according to how close it is to the user's daily caloric, macronutrient, and food group needs. Then the 20 best daily nutrition plans are selected. Then daily nutrition plans that contain repeating dishes or meals are filtered out. At the next step, weekly nutrition plans that contain repeating these meal sequences are filtered out. And finally, we have the weekly nutrition plan for the current and the next week for the specific user. Here you can see a screenshot of the application. And here I will try to show a very short 1-minute video that shows all the functionality of the application. So here you can see the log and sign up screen. The user can go to the sign up screen and complete the requested information here. Then an activation email is sent to them with an activation link. He can click on the link and then he is able to log into the application. The first thing is for the user to fill his profile. As you can see here, he selects all the attributes. And then the nutrition plan is presented to him for all the weekdays of the current week and the next week. On the left side, you can see the meals, and on the right side, the details of each meal. Also, there are some charts for weekly energy intake, nutrients and macronutrients, intake per day, and suggested values of the plan. And obviously an about page and the possibility for the user to log out. So the user can also update his profile, and then a new plan is suggested to him according to his new profile, and this is the basic functionality of the application. Sorry, I have to go back to the presentation mode. Okay, now I think that I have a couple of minutes to say very quickly a few things about the game that we are developing. It is an educational game for the Mediterranean diet. It is a life simulation game. The aim for the player is to choose appropriate meals and physical activities for a digital companion character. These Tamagotchi-like characters. Constant feedback is given to the player regarding the nutritional and healthy lifestyle value of their choices. And the aim of the game is education on key healthy dietary habits of the Mediterranean diet. Some further details. The player is responsible for the well-being of their digital companion. They should feed their companion at least 3 meals per day. They should lead their companion to work out at least once per day. And they should choose meal ingredients in different food groups. They should also choose healthy ingredients. This game was the design of this game was based on a game-based learning design model, and we used most of the factors present in this model. And we also added an extra feature called transfer to real life. Also, I would like to mention that this design procedure was presented recently in a conference. Now, here you can see a few screenshots of the game. The meal screen on the left, the character customization in the center, and a screenshot from one of the workouts. And here I have a very small video to show. So here you can see that it is lunchtime and the user selects the ingredients for the meal. According to the selection of the user, he's scored based on how healthy these selections are. And then he can proceed to verify his meal by choosing that the meal is ready. Also, I would like to stress that our main focus for this game are younger people, children. And so we have some checks so that they do not use many times during the day the application. Here you can see the food groups that were selected. And also the user gets some rewards for choosing healthy foods and meals during each day and also on a weekly basis. Thank you very much for your attention, and also thank you for the invitation.
Thank you very much, Lazarus. There is no question in the chat, but I have one question that I think it might be shared among or between Perla and Lazarus. It's firsthand is how artificial intelligence is helping switch to healthy application, in this case to empower families, members in following the dietary plans, but then as well, in both cases we are targeting young people, young children, and then my question here was, how can we balance the use of the digital tools, how they can be accommodated within their daily life, because also there's this controversy, on decreasing or not fostering, that people are engaged or are attached, to digital support. So how we can maybe teach them to use it in a manner, an appropriately manner, to get the necessary feedback or empower them, but then not too much. Let's see. So I don't know, maybe Lazarus, if you can start, and then Perla, if you want to add on top of it.
Yes, sure. Yes, this is a great question, actually, and we faced these issues, let's say ethical issues, also in the artificial intelligence part, and in the switch to healthy application, the artificial intelligence part is only working for the adult population. So for the children, we have directly menus created by nutritionists. So we didn't use the AI there. And also in the game, we thought about that because these Tamagotchi kind games can be used by a user during the whole day. So we have some checks there. And first of all, when the user chooses a meal, for example, his breakfast, after that, there is no functionality in the application until the next meal. And during this time, if he tries to access the application, he gets a message that it is not yet time for your next meal. Please come back in a few hours. Also for the children, we don't let them use the application during the morning hours where they are supposed to be in the school. So they again get a message that please come back for after your a few hours for your lunch or something like that.
Okay. No, Perla, if you want to.
Yes, of course, we should hope for parental control as well, but also maybe the app could be useful to provide also contents to apply in terms of practical games and recipes. So, for example, not only contents to be read in the app, but also activity to try with the family, for example, cook recipes together. So yes, translate the app in a more practical context, I think, in my opinion.
Okay. Thank you very much again, both of you, for presenting the current developments in the different projects. I think if there is no other questions, I think we can finish the sessions. Also, thank you very much all the people attending the webinar. You've seen a little bit of bits of results from MED4Youth. Switch to Health is still running, and you can follow as well to see how interventions goes. The presentation will be available in the web page of the MED4Youth, and also the recording. But I think you will also get through email. I hope it has been an interesting event. And again, if you have any additional questions, you can. It's already in the chat. You can type to info@med4youth.eu, and then we redirect any additional question that you might have. Thank you very much, and have a nice day. Bye-bye.